spinal cord. And how do I know that this is thoracic spinal cord?
Lateral.
Okay, you have a lateral horn. Okay, lateral horn is only present in the thoracic spinal cord from T1 to about L2. It actually extends into L2. Okay, but the reason you have a lateral horn is because the ventral horn consists of a medial nucleus, which controls axial muscles, and a lateral nucleus, which controls the appendicular muscles of the arm and leg. Well, in the thoracic spinal cord, you have no appendicular muscles. So the lateral nucleus, which is present during early development, the lateral nucleus, which occupy this space right here, is gone. Okay? They die. They're programmed to die because they fail to reach a target. Now, if you were to take a supernumerary limb bud and you were to implant it into the side, the lateral side of the chest wall of a vertebrate during embryonic development, what will happen is that the lateral nucleus will become viable. They will not undergo apoptosis, okay, because they have a target that they can innervate. So in that case, you would have one side that does not have the extra limb, thoracic limb, will undergo degeneration or apoptosis, and then you'll be able to clearly see a lateral horn. But on the side where you have a limb bud, you're going to have the lateral nucleus, and the lateral horn is not going to be so obvious anymore because now you have a lateral nucleus occupying this space. Okay, does everybody understand that? Okay, so the lateral horn contains the preganglionic sympathetic neurons, and they will be located in spinal cord levels T1 through L2. Okay, and it's called the lateral horn. The nucleus is called the intermediolateral cell column. Intermediolateral cell column. Okay, so the intermediolateral cell column is the name of the nucleus, and the lateral horn is the name of the anatomical structure where they're located. Okay, they will give rise to myelinated axons that will exit through the ventral roots here, the ventral rootlets. The ventral rootlets will come together to form a ventral root. Okay, so they all come together to form a ventral root. So the ventral root is not directly connected to the spinal cord. The dorsal root is not directly connected to the spinal cord. What intervenes are these rootlets. So these rootlets emerge from a spinal cord segment. The spinal cord rootlets will extend from here to here, and all of this defines a particular spinal cord segment. Okay, there's no overlap. Like these rootlets do not overlap with these. So this is one spinal cord segment. This will be another one. Okay, so the rootlets come together as they exit the spinal cord where the motor neurons are located, and they form a ventral root. Now, when the ventral root merges with the dorsal roots are carrying sensory information toward the spinal cord. But the dorsal root also contains ganglion cells in the middle of the axon, and they form the dorsal root ganglion. The axons continue on their way, and then they break up and separate as rootlets before they enter the spinal cord. So the extent of the rootlets, the spread of the rootlets define a spinal cord segment. And some of the axons, the large diameter axons, will form the dorsal column, carry fine sensory information up the dorsal column to the spinal medullary junction, where they will form synapses. The leg fibers will ascend up the fasciculus gracilis. They will terminate in nucleus gracilis. Nucleus gracilis will send an axon fiber tract up the contralateral brainstem to form the lower half of the medial lemniscus, standing on the pyramid, right above the pyramid. And then the upper extremity will send their axons in to form the fasciculus cuneatus. Fasciculus cuneatus will ascend the spinal cord all the way up into the upper cervical level at the spinal medullary junction, and they will terminate into the nucleus cuneatus. The neurons of nucleus cuneatus will send a cross projection and add the medial lemniscus fibers to the top of the fibers that represent the leg. Okay, so you're going to have the top of the medial lemniscus will carry arm fibers. The bottom of the medial lemniscus will contain the leg fibers. So here you have a headless, half-human representation in the medial lemniscus standing on the pyramid. Okay. Now, as that medial lemniscus ascends up the spinal cord, it's going to rotate laterally such that the legs are going to end up lateral in the lateral position. The arms are going to be located in the medial position. When the medial lemniscus gets up to the level of the rostral pons, head is going to be added from the trigeminal thalamic pathway. Okay, so the fine sensory nucleus of the trigeminal system will add to the medial lemniscus medially, where the head representation is added to the arm and the trunk. So you're going to have and leg. So you have lateral to medial. You have leg, then arm, then trunk, then arm, and then the trigeminal system will add the head. So now you no longer have a headless half human. Now you have headless half human with a head added from the trigeminal system. And then that projection will continue to rotate so that the legs are going to be twisting up into a dorsal lateral position, and the head is going to be in the ventral medial position as it enters the area of the ventral basal nucleus. So the arm, trunk, and leg fibers are going to terminate into the ventral basal portion called the ventral posterolateral nucleus. And the head fibers are going to terminate in the ventral posteromedial nucleus. Okay? And then the thalamic nuclei of the ventral basal complex are going to send a topographic projection, and they're going to terminate into the somatosensory cortex and the postcentral gyrus of the brain. Okay, so the leg fibers are going to end up medially, right above the corpus callosum. The arm fibers are going to end up in the dorsolateral part of the somatosensory cortex. The leg are going to end up, the head is going to end up in the lateral portion of the somatosensory cortex, and then the tongue and the pharynx and regions are going to end up inside of the lateral fissure, okay, in an area called the insula. Okay, so there's an ordered topographic projection from the inside of the oral cavity to the mouth, to the head or face, arms, trunk, and leg. An orderly progression as you go from the interior of the lateral fissure, okay, and you come out along the postcentral gyrus and the somatosensory cortex, and as you go lateral, you have the head is lateral, and then you have the arm, dorsolateral, and then you have the trunk, dorsal, and then medial is going to be your legs. So that's how the topographic projection gets there. Okay, so other things to know is the lateral horn consists of the intermediate lateral cell column. It's at the level of T1 to L2. These are going to be preganglionic sympathetic neurons. Okay, preganglionic sympathetic neurons. Their axons are going to enter the ventral rootlets. They will enter the ventral root, and they will even travel for a very short distance in the spinal nerve. The spinal nerve is where the ventral and dorsal roots come together. So you have a motor component ventrally and a dorsal sensory component. And then the preganglionic sympathetic neuron axons are going to travel for a short distance in this, where the spinal nerve is formed, and then it's going to bypass the dorsal ramus that will innervate the back. It'll bypass the gray ramus of the autonomic nervous system, and it'll leave through the white ramus. Okay, so the white ramus will contain myelinated axons of the preganglionic sympathetic nervous system. Those axons will terminate in the sympathetic ganglion. Okay, the sympathetic ganglion cells will receive synaptic input from myelinated axons originating in the lateral horn or intermediate lateral cell column, and the axons that emerge from the sympathetic ganglia are going to be unmyelinated. And because they're unmyelinated, their axons are going to travel in a fascicle of axons that they do not look white. Okay, so myelination gives the nerves the white appearance. So instead, this efferent axon bundle is going to consist of unmyelinated axon. And it's going to have a gray appearance. Okay, so this is called the gray ramus. The myelinated axons form the white ramus. The unmyelinated axons of the postganglionic neurons of the sympathetic trunk. They will send unmyelinated axons. Some will go to the skin to innervate the erector pili muscles, sweat glands, and other structures in the skin. Other axons will bypass the ganglion and enter with the splanchnic nerves to innervate structures in the abdominal cavity and the thoracic cavity, okay, and other structures. This diagram shows you the intermediate lateral cell column of the preganglionic autonomic neurons, okay, of the sympathetic nervous system. So these are preganglionic sympathetic neurons. They are cholinergic neurons. They will send their axons through the white ramus. They will innervate target cells within the sympathetic ganglion. And the postganglionic neurons that receive input from the preganglionic neurons, in the case of the sympathetic nervous system, will release the neurotransmitter norepinephrine. Okay, so it's a different kind of neuron, different neurotransmitters used. So basically, the final output of the sympathetic nervous system is primarily norepinephrine as the final neurotransmitter that interacts with effector organs. And that norepinephrine can cause vasodilation or vasoconstriction in different parts of the body in such a way that if you need more blood to go to your muscle, then the sympathetic nervous system stimulates increased blood flow to the muscle. Okay, increased blood flow to the nervous system to control those muscles. But it will have it'll stimulate a decreased blood flow through vasoconstriction of the peripheral blood vessels. Okay, so that more blood can be devoted to the muscle rather than the skin. Now, why is that important? Why, under conditions of stress or danger, would you want more blood going to your muscle and nervous system as opposed to your skin?
So that you can react.
Pardon?
Can have a full reaction instead of a partial.
That's right, so you can give a stronger reaction. You need more blood to supply the muscles that you're going to need in case you have to run away. Okay, so, and that produces, also associated with that oftentimes is fear. But if you can't run away and you're stuck and you're trapped, it also provides you with the energy and strength to face your danger and fight. This is called a fight or flight response. You have the choice of either fighting or flighting if you are put in a stressful situation. Okay? In both cases, norepinephrine release is increased in the sympathetic nervous system so that your body prepares itself for that stressful environment, protects you, defends you. Okay. Now, the parasympathetic nervous system has the opposite effect. It becomes active when you are eating a meal, when you are in a relaxed state. Okay. You don't fight battles with the parasympathetic nervous system. The parasympathetic nervous system is what is stimulated when you are in your relaxed state. There's no danger. Okay. And it also is hyperstimulated when you are eating a meal. Okay, and participates and stimulates the digestive processes. It causes a release of salivary enzymes from your salivary glands. It causes a release of digestive enzymes in your GI tract. It'll stimulate the release of lipases and amylases and glycosidases and all sorts of nucleases, proteases from your pancreas, so that the different foods that you have digested can be broken down further into their monomeric units, which can be taken up by the cell as a nutrient. Okay? Digestion into the monomers occurs almost completely outside of the nervous system in your GI tract. If you cannot break down certain products in your digestive tract to their monomers, they typically cannot be absorbed by your GI tract. Okay? For example, if you have a dimer saccharide, disaccharide such as lactose is a good example, lactose sugar. Okay, lactose sugar is a disaccharide consisting of a glucose and a galactose monomer. Okay, it's a disaccharide, two sugar molecule. It is too big for the plasma membrane to take it into the cell, so it stays external in your digestive tract. Okay, unless you have a special enzyme to break that chemical bond. And the special enzyme that you need to break that particular bond is called lactase. Okay, lactase. It's a type of beta glycosidic bond enzyme that separates the galactose from the glucose. Okay, in this particular case, lactose sugar happens to be stuck together with a beta glycosidic bond, unlike most other carbohydrates. So it needs a special enzyme to break that bond. It's the same kind of bond, a beta bond, glycosidic bond, is the same kind of bond that is used to make wood. Okay? And most of us don't have the enzymes to break down wood. That's why we don't eat our chairs and tables and trees. Okay? And that's why you have trees and forests, because very few organisms have the ability to break down wood. Now, if nature decided that it wanted to break down wood and these organisms evolved that is able to break down cellulose relatively easily, we would lose all our forests. Okay, but instead it's confined to a particular species of bacteria that are located, they live in the gut of termites. Okay, so the termites, what the termites do is they chew the wood into make little tiny particles, and then the gut bacteria have and protozoa, gut bacteria and protozoa have enzymes that can break down the beta glycosidic bond so that the termite can then digest it in its digestive tract and absorb the galactose and glucose monomers. Okay, but humans don't have that, but they have lactose. They have lactase enzyme which breaks the beta bond. Okay, so that's got sidetracked a little bit. But anyway, so in the spinal cord here you have, this is the ventral median fissure. Okay, so in the ventral part of the spinal cord you have a fissure. The dorsal part you have a sulcus, and you also have a septum. The septum separates the right and left dorsal column. And then you have an intermediate septum that separates fasciculus gracilis, which is medial, from fasciculus cuneatus, which is lateral. Fasciculus gracilis carries sensory fibers from the leg. Fasciculus cuneatus carries sensory fibers for the arm. Okay, and the trunk is divided half and half between the two. And the head is added later on when you get into the brainstem.
So can you tell more about the blood supply?
Okay, the blood supply, there are two major sources of blood supply. One is called the ventral median artery. Okay, and then you have the dorsolateral arteries that supply the dorsal part of the spinal cord. But the ventral median arteries are the most important because they supply most of the gray matter, okay, and of the ventral half of the spinal cord. But the gray matter is where all the neurons and synapses are located. But you still need the dorsolateral supply to supply the dorsal columns, okay, and part of the dorsal horn. Okay, so this is the cross-section of the spinal cord.
So you don't want to talk about the radicular artery? No?
Radicular, no.
Okay. Just making sure.
Okay, so these are the vertebral arteries. So this is cervical. The cervical vertebra have holes in the transverse process to accommodate the vertebral arteries. And what this does is it enables the neck to rotate, and when the neck rotates, the neck vertebra rotate the vertebral artery with it. If you had the vertebral artery outside of the vertebra, what would happen is that if the vertebra rotated, it would pinch one side and relax one side, but it will pinch the other side and cause it to close. So this is a very good design. It allows the vertebral arteries to stay open no matter which way you turn your neck. And it's only the neck vertebra that have these transverse foramina. Okay, that's how you could tell the cervical vertebra from all the others, from the thoracic and lumbar and sacral vertebra. Okay. Now, surrounding the spinal cord, you have the meningeal layers. On the surface of the spinal cord, you have the pia mater, and then with a little bit of a space between the pia mater and the next connective tissue layer, which is called the arachnoid mater. Arachnoid mater has finger-like projections called trabeculae that connect the arachnoid barrier layer to the pia mater. And within that space will be filled with cerebrospinal fluid, and that cerebrospinal fluid acts like a shock absorber for the central nervous system. Not only the spinal cord, but also the cerebral hemispheres are actually floating in the cerebrospinal fluid. So the subarachnoid space is very important, and the arachnoid matter is what acts as a barrier to prevent it from diffusing out. Okay. Now, surrounding the arachnoid matter is a very thick connective tissue layer called the dura mater. Dura mater is very tough. It's like leather. Okay. So you have the dura mater and internal to the dura mater and attached to the under surface of the dura mater, you have the arachnoid mater. And then the arachnoid mater is filled with a space beneath it called the subarachnoid space, which is filled with cerebral spinal fluid. And the arachnoid barrier layer has attachments to the pia layer through these trabeculae of finger-like processes that attach to the pia surface. The pia surface is what follows the contours of the spinal cord and brain, okay, and brainstem. So the pia layer is actually attached to the surface of the brain. And the subarachnoid space is filled with cerebrospinal that surrounds the entire brain, so the brain is actually floating. Okay. So if I were to take my brain out of my head and flop it on the table, it would actually eventually collapse on its own weight. It won't collapse all together, but it will collapse somewhat. It won't retain its shape. It won't be as tall. It'll kind of collapse part way. And that's the consistency of the brain. It's very, very soft tissue. And the fixed brain I have that I will show you at another lab is fixed with paraformaldehyde or formalin, and it's fairly stiff. Okay, and you'll see that what happens there is that when you have a brain that is fixed, the arachnoid matter and dura matter usually will be collapsed on top of it. You will no longer see the subarachnoid space. So when a medical student is looking at any cadaver material and they look at the brain, the arachnoid matter and the dura matter are typically going to be collapsed onto the surface of the brain. But that's not what they will see in surgery. If they're going to be operating on a person's brain, they're going to see that the dura matter and arachnoid matter are not attached to the surface of the brain. They don't come into contact with the brain. They're filled with there's a subarachnoid space that fills the space between the two. And the spinal cord and the brainstem and brain are kind of housed in between all of that to protect the brain. So basically, it's like your head basically has its own safety feature, your safety helmet. In other words, your brain has its own car seat. Okay, so that's exactly what a car seat does. It protects the body, while your brain and cerebrospinal fluid protects your brain and spinal cord. Okay. Now, there's a procedure that they used to have where they drain the cerebrospinal fluid out of the brain, and in so doing, they will also drain the ventricular system. And then when they take X-rays, they can tell the difference between air-filled ventricles in comparison to the brain tissue. They can't do so with when the ventricular system is filled with cerebrospinal fluid because the density of the material is too similar. So they really can't see the ventricular system very well when they use X-ray unless they drain the brain. The unfortunate thing is that when they do that, the brain is very vulnerable to being injured because it's no longer floating and suspended in the cerebrospinal fluid. And it's also a very unpleasant experience. It's nauseating, painful, very uncomfortable. But at one time, this was the only way that they could actually look and see what the structure of the ventricles was. If you had a type of vascular lesion, or if you had an enlarged ventricle, such as in hydrocephaly, they would be able to readily see that using pneumoencephalography, which involves draining the brain of the cerebrospinal fluid. But now they have much better ways to do that. They have a procedure called PET imaging, positron emission tomography, which can be used without draining the cerebrospinal fluid out. And they also have magnetic resonance imaging, MRI, which enables the imaging technique to detect differences between white matter and gray matter, between ventricles and gray matter and white matter. You could see nerves, you could see blood vessels, and you could see the differences between gray matter and white matter within the cerebral cortex, for example. But it has enabled the neurologists to make better diagnoses. They were no longer limited to the pneumoencephalography procedure, which is a very painful and uncomfortable experience for the patient. So these medical advances have really helped a lot. And then PET imaging is a method of functionally activating a particular system and being able to see what parts of the brain become functionally activated during the time. It's based on the fact that whenever you have increased brain function in any part of the brain, the glucose utilization increases. Okay, so you have increased glucose uptake. If you mix the glucose with a glucose analog called fluorodeoxyglucose, so fluorodeoxyglucose then gets taken up by active neurons and active parts of the brain much more avidly than the lesser active parts of the brain. And the reason why you want to use fluorodeoxyglucose is because, for two reasons. One, the fluoride ion, or the fluorine atom in fluorodeoxyglucose is a positron emitter. Okay, it emits positrons. This is a natural property of this particular element. And the positron, as soon as that is emitted, it'll collide with some local electron. When an electron and a positron collide, it gives off, if it collides in this position, it'll give off 2 gamma photons in opposite directions. Okay, orthogonal to, or right angles to the collision of the positron and electron. And those 2 gamma photons can be detected by gamma photon detector systems. And that's what PET imaging does. PET imaging is able to detect amounts of physiological activity in a particular part of the brain. So it's a functional imaging system. Okay? It's a functional imaging system. For example, you could use the functional imaging system when you're, say, talking, and then you'll be able to see that motor cortex and the speech area happens to show increased blood flow. If you're just viewing something and looking at something, then visual cortex will show elevated levels of activity. If you're speaking, then the Broca's area will be activated, and the language area will also be activated, because you have to think about the words that you're going to be speaking, so both of these areas become activated. If you're just viewing something, then occipital cortex will be, if you're concentrating on viewing something, then the occipital cortex will, where his visual cortex is located, will be show increased activity. Okay, so in this particular model, this is a cervical vertebra, and you can tell this is a cervical vertebra for many reasons. It has a transverse foramina where the vertebral artery passes. It also has a bifid spinous process, which means it's forked. Okay, this only occurs in the cervical vertebra. So those are 2 characteristics, but in addition to that, you look at the shape of the spinal cord, okay, and in the spinal cord here, you have a very large ventral horn, okay, so you have a very large ventral horn, and you have a lot of white matter. This is characteristic of cervical spinal cord, okay? And the very large ventral horn is very prominent in cervical spinal cord, whereas in thoracic spinal cord, the lateral nucleus of the gray matter here will be gone, because the lateral nucleus innervates the appendicular muscles. Okay, and there's no appendicular muscles in a normal person with just a thorax, right? Unless he's got an arm coming out of the side. So the lateral nucleus usually is gone in the thoracic spinal cord. Okay, so here's a spinal cord. It is housed within the subarachnoid cavity where there is a lot of cerebrospinal fluid located, and it's surrounded by the arachnoid barrier layer, which is attached to the dura mater. So the dura mater gives very, very strong support and protection of the entire spinal cord. And the cerebrospinal fluid is locked inside of here. The dura mater is very, very tough and leathery, so it'll retain the cerebrospinal fluid inside of it. Okay, and the spinal cord is also protected further by a layer of adipose tissue with a lot of vascular supply of veins and arteries. But this adipose tissue adds added support to the spinal cord. So not only is the spinal cord suspended in the fluid, but that a fluid-filled sac, encased by dura mater, which is real tough, also is floating inside of a adipose tissue that is within the spinal cavity here. Any questions on the spinal cord? Okay. So cranial nerves, well, we can go through the cranial nerves. The cranial nerve 1 you don't see in the brainstem because it's associated with the forebrain. Okay, and it's the olfactory system, and it doesn't even form a regular nerve. Cranial nerve 1 consists of these tiny fascicles, numerous fascicles that originate from the olfactory neurons of the olfactory epithelium located in the roof of the nasal cavity. Those axons collect to form fascicles, and they pass through the ethmoid bone into little tiny openings called foramina. So the olfactory foramina allow the olfactory neurons to send their axons into innervate the olfactory bulb, which is located right above the nasal cavity. Okay, so these axons just have to travel a very short distance. These axons will travel through the cribriform plate, okay, and they will innervate the olfactory bulb, which is located right above them. And each olfactory neuron has a particular odor and specificity. Okay? It's very particular odor and specificity, and will respond only in the presence of a particular odor. Okay? And somehow the brain is able to interpret all of that information to give you your sensation of smell whenever you smell something. Okay, so cranial nerve 2 is the optic nerve, and it has several targets. The optic nerve has one target in the hypothalamus, and it'll innervate the suprachiasmatic nucleus, which is responsible for assisting in waking you up in the morning. Okay, wakes you up in the morning. And then the bulk of the axons emerging from the retina will form the optic nerve. And the optic nerve will project to the optic chiasm, and the optic chiasm is a place where some of the optic fibers will cross the midline and others won't. So the medial optic fibers cross the midline. The medial optic fibers are connected to retinal ganglion cells that are seeing the lateral visual field. Okay, the lateral visual field. And they will cross the midline, okay, and in such a way that the entire right visual field is going to be processed by the left optic tract and all the targets of the left optic tract. Okay, so right visual field is going to be processed by the left optic tract. And similarly, the left visual field is going to be processed by the right optic tract. And the optic tract main target is the lateral geniculate nucleus. A minor target would be the superior colliculus. Okay. Anyway, so cranial nerve 2 is the visual, is the optic nerve, okay, and it's responsible for your ability to sense the environment visually. The cranial nerve 3 is the oculomotor nerve, and it controls 4 extraocular eye muscles, 5 actually, if you can include a levator palpebrae. But it also is able to control the contraction of the superior rectus muscle, okay, inferior rectus muscle, inferior oblique muscle, and the medial rectus. So all of these muscles are controlled by the oculomotor nerve. Okay, then you have the trigeminal nerve exiting the side of the pons, and the trigeminal nerve is responsible for fine sensory, pain and temperature information, and proprioceptive information for the face. The fine sensory and proprioceptive information is going to be relayed to the ventral basal complex in a place similar to where the somatosensory projection from the spinal cord also ends up. Okay, it ends up in the somatosensory thalamus, which is called the ventral basal complex, which includes the ventral posterolateral nucleus and the ventral posterior medial nucleus. All right, there's a motor component of the trigeminal nerve as well. The motor component of the trigeminal nerve is responsible for controlling the muscles for chewing. Okay, muscles for chewing. And the sensory component is for the different sensory modalities for the somatosensory modalities, including pain and temperature, okay, including fine tactile sensations, and including proprioceptive signals that can be detected. The pain and temperature pathway is going to be processed in what is called the descending or spinal nucleus of 5. And those axons in that system will eventually end up being processed in the ventral basal nucleus of the thalamus, along with the anterolateral tract. The fine sensory pathway is going to be carried by the trigeminal nerve, and the fine sensory pathway is going to terminate into the main sensory nucleus of the 5, which will be processed also in the thalamic nucleus. And that'll go from the, that'll add the fine sensory to the face, to the arm and leg that is carried by the medial lemniscus. So the trigeminal thalamic fine sensory pathway is added to the medial lemniscus pathway for fine sensory information. That gets relayed to the somatosensory cortex. At the caudal pons, you have 2 cranial nerve nuclei. You have the abducens nucleus that abducts the eye through the abducens nerve, and you have the facial nucleus that controls the muscles of facial expression. The abducens nucleus exits medially at the pontomedullary junction. The facial nucleus exits just lateral to that. And then you have, so that's cranial nerve 6, abducens, cranial nerve 7, facial, and cranial nerve 8 happens to carry the fibers from the auditory and the vestibular system. And we'll talk about the cranial nerve 8 when we talk about the auditory and vestibular system after the midterm. Cranial nerve 9, 10, 11, and 12 exit different parts of the medulla. Okay, so cranial nerve 12 exits medial to the olive. The rostral part of the medulla has this red little structure here called the olive. Looks like it has the shape of an olive. And cranial nerve 12 is the only cranial nerve that has axons that exits medial to that. Okay, so that's cranial nerve 12 right there. 9, 10, and 11 have axons that exit lateral to that. Okay, and exits the lateral part of the brainstem. So cranial nerve 9, cranial nerve 9 is the glossopharyngeal nerve, and it carries information for things like taste, for the posterior 1/3 of the tongue. It carries, it controls the reflexes that are associated with swallowing, the swallowing reflexes mediated by cranial nerve 12. It has carries fibers for the inferior salivatory nucleus, which controls the secretory activity of the parotid gland. Okay, and so that would be the glossopharyngeal nerve, and then the cranial nerve 10 also exits laterally, and this is called the dorsal. This is called the vagus nucleus or vagus nerve. Vagus nerve is a wandering nerve that carries taste from the epiglottis region, and it is also your visceral motor nucleus for representing the parasympathetic nervous system, for the entire part of the digestive and respiratory and cardiovascular systems of the thorax, and the upper and middle abdomen. Okay, so in the digestive system, it covers the entire area from the epiglottis all the way to the descending colon. Okay, it ends at the beginning of the descending colon. So the transverse colon is the last part innervated by the vagus nerve. Okay, but it controls the functions associated with digestion and the release of salivary enzymes by the pancreas and the gallbladder. It releases bile from the gallbladder. It also controls, regulates digestive enzyme secretions in the stomach and the churning of the stomach, and it ties in with the enteric nervous system as well to control the digestive processes. Let's see, what else is there? Okay, so the cranial nerve 11 is called the accessory nerve, and the accessory nerve has its origins in the brainstem called the nucleus ambiguus. And because it's called the nucleus ambiguus, you ask a person where the nucleus ambiguus is, and they kind of shrug their shoulder and they say no. Well, the muscles that are required to say no includes the sternocleidomastoid muscle, and the shrugging of the shoulders involves the upper muscle fibers of the trapezius. Those are controlled by the accessory nerve. Okay, so it's a good way to remember the function and structure of the accessory nerve. Its nucleus is called nucleus ambiguus. You ask where it is, and you go like this, and those are the exact muscles that are being controlled by the accessory nerve. Okay, now there is a component of the nucleus ambiguus actually that really controls the some pharyngeal muscles and the swallowing. Well, not the swallowing, that's the glossopharyngeal. Okay, so the accessory nerve, it does control some other muscles in the neck region as well. The last cranial nerve is cranial nerve 12. It controls the muscles of the tongue. And so when you stick out your tongue, if the tongue is bilaterally innervated right and left side evenly, the tongue should stick out straight. If the tongue deviates to one side, the right side or the left side, that suggests that there's a lesion either in the hypoglossal nucleus or the nerve on the side of the lesion, on the side that the tongue is pointing to. Okay, so the action of the stimulation of the hypoglossal nerve and nucleus is when the tongue is stimulated on one side, it'll push the nucleus or the tongue to the opposite side. If it's a lesion, the innervation of the contralateral nucleus and nerve is able to overcome the lesion side and push the tongue toward the side of the lesion. So the tongue points to the side of the lesion. So if you want to freak out your doctor, the doctor asks you to stick your tongue out straight, you stick it out crooked, and you don't do that. That's really mean. I'm just kidding. Okay, but I tell you things like this because it'll help you remember. Okay, so if I tell you something really stupid, you'll remember it. Okay, and that's one of the stupid things I like to tell students so that they will remember it. Okay, so if it's a question on the test, what is that stupid thing that Dr. Takayama must remember it? Okay, any other questions? Are there any questions? I don't know how much time we have left.
5 minutes.
5 minutes, yeah.
where
Sorry?
does 11 exit on the
What?
where does 11 exit on the model? Where's cranial nerve 11?
Right here
Oh, okay.
on the side. So you have 9, 10, and 11. Some of these fibers from the cervical spinal cord are part of cranial nerve 11. So you have 9, 10, and 11. So this is 7, 6, 7, and 8. 6, 7, and 8, and 9, 10, and 11, and then 12 is right here.
Okay.
Okay?
Thank you.